Yeah, thanks, Dirk, for this lengthy introduction. I would like to start out in thanking the organizers for providing me with the opportunity to give you a glimpse of what we do at the Helmholtz Center for Infection Research and also the Helmholtz Institute in Saarbrücken in the area of novel antibiotics. So why novel antibiotics? Here is a societal challenge. I guess this is highly recognized now by all the authorities, by the WHO, by the EU Commission. We do have a sincere problem with antimicrobial resistance, AMR, and a recent study by the UK government here indicates that by 2050 we will have approximately 10 million deaths caused by AMR. So that's of course an issue. Who are the bad guys in this game? We've just heard about the bright side of microorganisms. So the bad guys are the so-called escape microorganisms. These are the most multi-resistance organisms that occur in the clinics. Most of them are gram-negative species. This is also one reason, because of their double membrane. They are really difficult to treat, and they have a lot of tricks which in the course of their evolution, they use to circumvent antibiotic treatment. So we have all been used to the availability of antibiotics. They have mostly been developed in the 50s and 60s of the last century. And then there has been a significant void in development because the authorities and many people have thought that basically infection research does not need any antibiotics anymore. And only in the last 20-25 years it has been recognized that the microbes fight back and now have all the resistance mechanisms. And we try, by using medicinal chemistry, to improve the compounds which are on the market, which basically leads to the few antibiotic classes in use to be derivatives of what has already been known and not targeting new and innovative targets. So this is also mirrored by what we see in the clinic, just few examples of multi-resistant species on the rise over the decades. And at the same time, unfortunately, the pharma pipeline is really running low. You see the number of antibiotics approved per year, and the number of companies still in the game is unfortunately decreasing. So what can we do? Actually, we rely on the most traditional resource of antibiotics, which is natural products from other microorganisms. So this brings us back to the bright side of microorganisms. They can also be used to harvest their evolutionary potential, which has brought to life almost all of the antibiotics which are currently used in the clinic. Few examples are shown here. And I would assume that 70–80% of the audience has at least used one of these in their past. So we all profit from these antibiotics, which all stem from few classes of microorganisms and fungi, which have been a little bit overstudied in the last decades. And that's also one reason for the developmental void. These classes of microorganisms have been studied so well that we now over and over find the same compounds again, which are not worth further developing. So at the Helmholtz Center, we have focused now over decades on a ubiquitous class of bacteria, the so-called Muxo Bacteria. Why do we do this? Well, first of all, they are just the coolest microorganisms on the globe. So whoever doesn't believe this should talk to me in private. They can do a lot of things which you would never imagine a microorganism being capable to do. They can creep on even surfaces. They can prey on other microorganisms. They build such little tree-like structures in which they form spores. They have the largest genomes of any bacteria. And last but not least, and most important to us, during their processes, especially of preying on other microorganisms, they form antibiotics. And this is why we try to study them to figure out what kind of compounds do they make and can we use them for future application. So here is the work line that we usually do. We try to isolate these bacteria from soil samples. We collect them, we cultivate them, we extract them, and then we analyze them by chemistry, analytical methods, and also bioactivity methods to figure out if there is a new compound that was not known to us. So that may sound very traditional. What has changed in the last decade, or maybe last 15 years, is that we now put very much more effort on biodiversity. So we know that only less than 1% of the microorganisms present on the globe have been cultured in the laboratory. So there's an enormous potential to get them to grow in the lab. We've seen all the genomics technologies, which now tell us how do the microorganisms make these compounds, how can we influence this, and how can we learn from them what are mechanisms to improve these compounds. And this is also mirrored in the development of an in-house database which we've set up, which holds all the data around these compounds and the microorganisms. And last but not least, the center holds a production facility, which is a fermentation facility in which the compounds are then made at a scale that is required for development, and the compounds are put into a library. So my first example, I only have two today, so don't worry, is from a real new species of a Muxo Bacterium, Cystobacter isolate, and this came to our attention because this isolate produced a compound, which is shown here, which is completely novel, which showed promising activity against the earlier mentioned escape pathogens, just with unfortunately no activity against Pseudomonas. But all the other escape microorganisms were hit. This is why we took the whole route to elucidate the structure, and we were happy to see this is a completely novel chemical scaffold. Now, the next question in such development is, what does it actually target? Is it specific? And here comes the new route in this area of research. We then look at the genome of the producer strain. We try to figure out how does it make this compound. We call this biosynthesis. So actually these microbes are sometimes the much better synthetic chemists. They do not use anything else than just water equals media, just a temperature around 30 degrees. So that's very advantageous. And they also tell us their story. What kind of enzymes do they make? And that has become a very important issue recently. They also tell us how do they protect themselves from this antibiotic to work. So they have to have a self-protection mechanism. And if you figure out what the self-protection mechanism is, this sometimes tells you what the actual target of this compound is. In this case, we found some indication in the biosynthetic gene locus for gyrase being the target. Gyrase, a highly validated target in antimicrobial therapy used by the fluoroquinolones. And we could then prove that indeed DNA gyrase is the target of this compound class. So this made us even more interested. So we went back to our strain library. It's a worldwide unique collection of more than 9,000 Muxo Bacteria. And using the database, we now can in silico ask the question, do we have other producers of this compound class? And we found such a number of them, looked at all of them in depth and recharacterized their bioactivities. And it was really gratifying to see that we then found also a derivative of these compounds which is active against Pseudomonas. And using this compound, we've then gone into a development which has in the meantime been funded by the German Center for Infection Research and also the Innovative Medicines Initiative. They have a part which is called ENABLE, European Gram-Negative Development Engine. We received some funding there to now go into the preclinical development which is required to get such compound into a stage where it can approach, hopefully, phase 1. So here just some of the data. I don't want to bother you with details. The compounds are highly active on the escape pathogens, and they show little signs of cytotoxicity, and there's a lot else that you need to study for application. So the whole idea is biotechnological production, total synthesis, and semi-synthesis have to go together to now improve the profile and the potency of this compound. To sum this part up, we do have a novel class of antibiotics here. What I have not mentioned yet, they show no cross-resistance with fluoroquinolones, which is highly important. So in case we could develop them, this is a validated target. But the species which have become resistant to the fluoroquinolones already are still sensitive. Here's my second example. Now we go back to the very old class of antibiotics, the so-called griselimycins. They are known since the 60s, but they were dropped in pharmaceutical development, although they show superb activity against Mycobacterium tuberculosis. So why were they dropped? They were simply not plasma stable. As soon as you put them into an animal, they degrade and the compound's not of any use. So together with our colleagues at Sanofi, or former Aventis company, we have moved into a project to figure out what actually causes this plasma instability. And we found a metabolic hotspot, which is right next here on this proline. And if you kept this, and this has been done by total and semisynthesis, you can achieve a stable compound. So this program has been run with Sanofi. A lot of chemical derivatives were generated, and the take-home message is we now have an improved compound, which is orally bioavailable, which has highly increased metabolic stability. And to make the name even more complicated, we now call this cyclohexyl griselimycin. So I will continue with griselimycin. So what is so special about griselimycin? In animal models, which have been performed in Jacques Grosset's lab, who invented the first TB animal models, they show a very nice dose response. Take-home message here is the higher the dose of the compound, the less tuberculi you find in the mice. But even more importantly, this is a treatment with the typically used triple therapy in TB, which combines 3 antibiotics. And usually in human therapy, you need a couple of months for treatment. In mice, you can achieve clearance in this triple treatment within 12 weeks. So when we replace any of these 3 known antibiotics with griselimycin, we can achieve clearance within 8 weeks. This is already important in itself, but the most important issue here is no cross-resistance with any of the new antibiotics. And you all have heard about MDR and XDR TB species, which are resistant to actually everything that is used in TB treatment. So we really need antibiotics that have new targets and do not show cross-resistance. So we then asked the question again, what is actually the target of this compound? We went back to the genome of the producer, which is a Streptomyces, sequenced it, figured out how this compound is made, which actually also helped to figure out how to derivatize this position, which is the hotspot. And we then found a copy of the so-called DNA polymerase sliding clamp in the biosynthetic gene cluster, indicating that this may be the target. So what is the sliding clamp? The sliding clamp is a ubiquitous enzyme that we also have, but in microorganism it looks a bit different. It's a protein, not an enzyme, actually. It forms a dimer and wraps around DNA. And only if this dimer wraps around DNA polymerase, and also DNA modifying and DNA repair enzymes can attach to this dimer. And only then you have DNA repair, and only then you have DNA polymerization. So this sounded like a highly innovative target. So we then looked whether in Mycobacteria resistance could occur. And this is shown here, done in Mycobacteria smegmatis and bovis. And they do evolve resistance. That's something all bacteria do. So resistance is not a matter of if, it's just a matter of when for all antibiotics. What is important is the frequency of resistance. So the frequency of resistance here is very low. And if the bacteria evolve resistance, they become basically sick, so their growth rate is becoming slower, and they have a huge discrepancy here between growth rate of the wild type, and they become sicker the more resistance they get. So this is 5 microgram per mil, resistance is 40, so basically there's a big disadvantage for the bacteria to evolve this resistance. We then, of course, asked the question, what happens on the genome scale for them to become resistant? To figure this out, we evolved M. smegmatis mutants stepwise in the lab to become resistant to griselimycin, and then sequenced those species which had acquired the resistance. And the outcome of this study was heavily surprising. because we did not see just a single gene mutated or promoter mutated. These strains amplify a large region around the origin of replication of their chromosome, which is almost 15 kb in size, and that also explains why they become sick. And we went one step ahead and checked what happens if we take away the antibiotic pressure, and then they lose all these multiplications. So basically, resistance is reversible. Now you may say this has only been done in smegmatis. We have also done this for Mycobacterium tuberculosis by looking at isolates which came out of mice infected with MTB that were treated with sub-inhibitory concentrations of the compound, and they gave the same results. So we then tried to get a crystal structure of grisilimycin with this target that's shown here. This is exactly the binding site which is known for the interaction with DNA polymerase and the sliding clamp. And also the DNA repair enzymes bind to this region of the protein. So what you can also see in this animation, this is one of the, one would basically call it a miracle compound, which inhibits protein-protein interaction on the target, and the binding coefficient is 10 to the minus 11. So basically, once this compound sits on the sliding clamp, it will not go away. To sum this up, grisilimycins really show outstanding in vivo activity, no cross-resistance, very limited and reversible resistance is occurring, and it blocks DNA polymerization by inhibiting the protein-protein interaction of DNA-N with the polymerase. So both of these compounds are still in late preclinical or preclinical phase. Of course, our aim is and has to be to move these compounds into real clinic, but I would like to point out these are not antibodies where there is a clean-cut path forward into the clinic. These are small molecules where there's a lot of toxicity, etc., which has to be studied. So my last two slides, just to, as we've mentioned earlier, this symposium should also encourage further interaction. This is an interdisciplinary research between chemistry, biology, pharmacy, and also chemical biology. So our strain library yielded a lot of interesting chemical biology tools which are of use in various other applications. We are happy to share this with you. And we also, within the topic at HZI, have the capabilities to make these compounds either by chemical synthesis or by fermentation. So here are the PIs in topic 3, which are located at Helmholtz Center in Saarbrücken, and our partners at the Leibniz University, who focus all the way on this chain from isolation of bacteria to their development, chemistry, formulation. This is what we do. And of course, special thanks goes to my group at the Helmholtz Institute and our collaboration partners. And I'd like to thank you for your attention.